Hello everyone, and welcome to the latest MDPI webinar on biomolecules, which is entitled Understanding the Potential of Host Defense Peptides in Treating Cancer. Thank you very much for joining us today. This is the second webinar of a series based on special issues and sections from our Biomolecules journal. So if you're interested on submitting your paper to the special issue that is related to this webinar, just check the webinar's website. Our chairs today are Dr. Melanie Coombs and Dr. David Hoskin. And we will have three presentations, starting by Dr. Hans Vogel, then Dr. Valerie Booth, finally Dr. Haney. After each speaker presentation, we will have a short Q&A session during which we will address some of the questions that are submitted by live online audience. If you're joining us live, before we get started, I will share some of information for you, for online viewers. So at the bottom of your screen, you will have both the Q&A and chat boxes. So for the Q&A, that will be for the Q&A session. So please start thinking about your questions and submit them at any time during the webinar in the Q&A box. Very important, not in the chat. And then you submit it there, you click send. And as we have more than one speaker, it is also very important that you address your question. So mention the name of the speaker together with your question. On the other hand, you can use the chat box, for sending your comments about the webinar, and it should be sent to all panelists and attendees. Check that you're sending it to all panelists and attendees, or to all panelists, wherever you wish. You can also log into your Facebook, LinkedIn, or Twitter accounts during the webinar to post updates or send tweets about the event. And I would like to remind all of you that this webinar is being recorded and that the recording will be available soon at the webinar's webpage. Also, certificate of attendance will be available on demand on the biomolecules.webinar at mdpi.com address. Now I'd like to introduce our chairs. So Dr. Melanie Coombs is a principal investigator at Acadia University in Canada. Her research interests include the investigation of the anti-cancer and immunomodulatory effects of natural products, such as phytochemicals and antimicrobial peptides, both at a cellular and a molecular level. A recent focus has been to examine the antimicrobial peptide mastoparan from wasp venom and derivative of it, to study the mechanism of action on killing cancer cells, as well as effects on immune cell function. Dr. David Hoskin is professor at Dalhousie University in Halifax, Canada. He found that a fragment of milk protein known as lactoferricin triggers the death of cancer cells obtained from a wide range of human malignancies, including breast, colon, and ovarian tumors, and various leukemias and lymphomas. Recent studies from the Hoskin laboratory have focused on the anti-cancer activity of pleurocidins. Dr. Hoskin is currently working on methods to deliver these anti-cancer peptides directly to cancer cells. So now I hand over the microphone to our chairs and enjoy the webinar.
Hey, hello everyone. What we're going to do here is Dr. Coombs and I will give a brief introduction to the use of host defense peptides as anti-cancer agents, and that will then be followed by talks from our three speakers. So this is an outline of the introduction. Talk briefly about cancer treatment challenges, some of the properties of different host defense peptides that are effective against cancer cells, the potential for some cancer cells to develop resistance to these peptides, and how anti-cancer host defense peptides can be optimized for systemic administration. So why do we want to study host defense peptides as potential cancer therapeutics? Well, as you probably know, cancer is a major and growing health problem, both in the developed and developing world. Cancer development is linked in many cases to chronic inflammation, and that results in changes to the DNA, mutations that lead to primary tumor growth and eventually spread of the tumor to distant sites in the body. Current treatments for cancer, particularly cancers that have spread involve use of chemotherapy, chemotherapeutic drugs, but these drugs come with problems. There's toxicity associated with these drugs, and that results in some patients not being able to complete their course of treatment or receive an effective dose. And indeed, some chemotherapeutic drugs cause secondary malignancies. A big problem is cancer cells that develop resistance to these drugs and then can no longer be affected by them. And in terms of metastasis, an important target is cancer stem cells, which originate new tumors, but these stem cells are typically highly resistant to standard chemotherapy or radiation therapy because of their low rate of division. And of course, we want to deal with metastasis, which is a very complex process, as illustrated in this panel. So, host defense peptides are widely distributed throughout nature. We find them in many different species, everywhere from insects to reptiles, amphibians, fish, mammals, birds. We find them everywhere. Not all host defense peptides have anti-cancer properties. What we've done is list here a few that are well known, have been well characterized for their anti-cancer activities, and the two in red, lactoferricin from cows and pleurocidin-like peptides from Atlantic cod, are two that my laboratory and Dr. Coombs' laboratory have worked on. More recently, Dr. Coombs has started to study mastaperin, which comes from wasp venom. What I want to point out here is the importance of certain amino acids in these host defense peptides. These peptides are typically less than 40 amino acids in length. They're cationic, so they have a negative, a positive charge, and that's typically due to arginine and lysine residues. And they also have bulky amino acids, such as tryptophan, which are involved in membrane insertion. So there are a number of models that have been proposed for the selectivity of host defense peptides for cancer cells. The common feature of these models is that the net negative charge that is typically present on the surface of cancer cells due to their elevated expression of anionic or negatively charged molecules such as phosphatidylserine results in an electrostatic interaction between the host defense peptide and the cell membrane, after which the bulky hydrophobic amino acids insert in the lipid bilayer and disrupt the lipid bilayer, depending on the concentration of host defense peptides attached to the cancer cell. That disruption can range anywhere from relatively few well-structured pores, as shown in the barrel stay model, to close to complete disintegration of the cell membrane, which is seen in the detergent model. So the membrane of the mechanism of penetration depends on the host defense peptide structure, its length, and also its degree of amphipathicity, or how many of those bulky amino acids are present in the molecule. Now, there are other factors that contribute to the selectivity of host defense peptides for cancer cells. Cancer cells tend to have a higher transmembrane potential, so that provides an electromotive force promoting peptide insertion into the cell membrane. The actual structure of the cell membrane is different. One finds more cholesterol in the cell membrane of healthy cells, and that makes those membranes more rigid, thus making it more difficult to destabilize compared to the cancer cell. Cancer cells typically also have a greater surface area, shown here, because of the presence of abundant microvilli. So that would allow then for the interaction of greater molecules, greater numbers of peptide molecules with the cell membrane. So these are some factors then that account for the selectivity of host defense peptides for cancer cells over normal healthy cells. This is an illustration of a eukaryotic cell showing some of the reported interactions of host defense peptides with different organelles as well as the cell membrane. And I just want to point out that all anti-cancer host defense peptides interact with the cell membrane, and depending on their mode of action, may enter the cell and then interact with mitochondria, the nucleus, enzymes such as phosphatidyl 3 kinase, and other structures. In terms of the killing of cancer cells by host defense peptides, there are two mechanisms. There's a direct cytotoxic effect, which results from membrane lysis, and an example of this would be the pluricidin NRCO3 interacting with a breast cancer cell. This electron micrograph shows the extensive pore formation and resulting in high levels of cytotoxicity to those cancer cells. In contrast, other host defense peptides don't cause extensive membrane damage, but they cause sufficient membrane disruption to allow that peptide to enter the cell and then target internal structures such as mitochondria. And this is the electron micrograph showing the mitochondria of lactoferrin-treated leukemia cells. And you can see that the mitochondria have become swollen, and that results in triggering the mitochondrial path of apoptosis, which can be blocked with different caspase inhibitors. Importantly, cancer cells that become chemo-resistant remain sensitive to host defense peptides because of their mechanism of action. Chemosensitivity, or rather chemoresistance, is typically the result of expression of drug efflux proteins in the cancer cell, and that mechanism of resistance has no effect on a peptide which is going to damage the cell membrane. In fact, damage to the cell membrane may account for the ability of some of these host defense peptides to enhance chemotherapeutic cytotoxicity. So one could predict then combining host defense peptides with conventional chemotherapy would give you an equally an effective anti-cancer activity with a lower dose of chemotherapeutic drug, thereby reducing unwanted patient toxicity. In terms of thinking about these molecules as therapeutic agents, it's important to determine the smallest possible structure which gives the desired effect. That's from both an economic standpoint. It's less expensive to synthesize smaller molecules, and it's easier to develop delivery systems for smaller peptides for patients. So, for example, for lactoferrin, this 6 amino acid sequence is the minimal sequence for a cytotoxic effect, but only if that very short peptide is enclosed within a liposome that allows that peptide to gain access to the cytoplasm of the cell. So once within the cell, that very short peptide will trigger caspase-dependent apoptosis within the cell. Now, that peptide, remember, if it's not in the liposome, won't harm the cell. But if this is another illustration of the importance of getting those short peptides actually into the cell. If one couples with a glycine-glycine linker, a membrane penetrating sequence of amino acids consisting of 7 arginine residues, that dual structure, that hybrid molecule, now becomes highly cytotoxic to cancer cells, looking at different T and B leukemia and lymphoma cells, but remains harmless to normal activated peripheral blood lymphocytes. Okay, Melanie, on to you.
All right, so we've also looked at some of these host defense peptides and how they can have an effect in a mouse model. So on the left here, we see in an immune deficient mouse, example of B lymphoma cells that were injected. And we see here that in treating them with the lactoferrin B, we improve survival of these mice over time in comparison to the saline treated mouse. On the right, it is an example of breast cancer cells injected in the flank of an immunodeficient mouse. And looking at the tumor volumes, we see that 3 injections intratumorally of these pleurocidin-like peptides significantly reduced tumor volume compared to a buffer solution that injected into these tumors. So we see that in both of these mouse models, in a hematopoietic type of cancer as well as a solid tumor model, that these peptides given intratumorally and in the intraperitoneal cavity in the example of the lymphoma, that they significantly improve survival as well as decreasing tumor volume. So another way that these host defense peptides work have been shown to be through producing an immune response. And immunotherapy is important for the effective treatment of cancers. And if we look here on the left, there's an example of what can happen when you can have an immune response to cancer cells. So in this example, we have a tumor, and then the presentation of tumor-specific antigens by antigen-presenting cells to a local draining lymph node, presenting those antigens to T cells, activating a T cell specific response. And hopefully those T cells would then go and kill those cancer cells. What happens often is there's an upregulation of T regulatory cells that inhibit those specific T cells, as well as immunosuppression that occurs due to upregulation of molecules on the surface of those cancer cells. There have been several reports indicating that some HDPs are able to inhibit certain aspects of the immune response, so inhibit the Treg cells, and then also increase an immune response with a specific cytotoxic T cell response and improving natural killer cells as well. There have been studies looking at some host defense peptides that produce a long-term T cell response, giving a specific immune response, and then other peptides that have shown that if you combine a peptide with some cytokines like IL-12, it can improve survival long-term in tumor-bearing mice. So if we look at a couple of these a bit closer, this is an example of looking at the response of the peptide LL37 and how it can induce apoptosis in T regulatory cells. In here, they're looking at tunnel staining, and so when there's an increase in this percent of tunnel positive cells, there's an increase in DNA fragmentation and therefore apoptosis. And so we see an increase in apoptosis in the T regulatory cells, so the CD4 positive, CD25 positive, and the FOXP3 positive cells, whereas there is a very little DNA fragmentation occurring in CD4 positive cells that don't have those CD25 or FOXP3 on them. And this is true in non-stimulated as well as stimulated T cells, both with that LL37 peptide. So overall, if we know that T cells are able to produce a specific immune response to our cancer cells, they can sometimes be inhibited by T regulatory cells. And this peptide, LL37, is able to inhibit that inhibitor of the T cell response. Another example of an in vivo situation where you have peptide treatment, and an example of showing how the immune response is effective here is this LTX-315, and it's an example of melanoma in mice. So they showed here in this paper that there's an increased survival in mice that have been treated with LTX-315 and after a re-challenge intradermally. So these mice were cured of the tumors with LTX-315, and then they re-challenged them intradermally, and they saw an enhanced survival, suggesting that they had some protective immune response to this type of tumor. They also re-challenged intravenously with these melanoma cells, and they found that there was an increased protection and a decrease in the amount of tumors that formed in the LTX-315 treated mice compared to controls. When they looked in the tumor sections after treating with the peptide compared to control-treated mice, they looked at a few different cytokines, and this is one example. They looked at IL-6 production in the tumor, and so by mRNA expression in the tumor, they found that there was an increased amount of IL-6 4 hours after the injection of the peptide, and they found in the serum level the mice, the IL-6 increased as well. They further tried to examine how this peptide was possibly enhancing this protective immune response, and they looked at the production of HMGB1, which can be released from cells that are dying and kind of produce a danger signal. And they show that around 10 minutes and then 30 minutes, they have an increased amount of HMGB1 in the supernatant compared to the lysate with the control. So these are a few examples of suggesting that this peptide is important in protecting producing a protective immune response in these melanoma mice. And they have looked further, so this is a more recent paper looking at some melanoma tumors in patients. And so they have, they're trying to identify whether or not there's an immune response in these patients with treatment with LTX-315. And so they found that by looking with immunohistochemistry for CD3 positive and CD8 positive cells, they see an influx of T cells after treatment with the LTX-315 compared to before treatment. And they show here 2 patients, the baseline levels of the CD3 positive cells in 2 different patients, and then also the CD8 positive cells, indicating that there's likely cytotoxic T cells moving into the area of these 2 patients. They also show this for sarcoma tumors as well. So we're starting to see these host defense peptides being used as treatments in patients. One important question would be, are these going to produce resistance? Are these cancers going to be resistant to the peptide treatments? And so this is an example where we took breast cancer cells and treated them for a long period of time with increasing concentrations of the peptides, either NRCO3 or NRCO7. and we tried to make them become resistant to them. So looking for whether or not cancer cells can become resistant to these peptides. And we do see that they become resistant over time. And so they do structurally or morphologically look very different than the parent cells. But they are still able to be killed by other chemotherapies. So cisplatin and docetaxel and tamoxifen are still able to effectively kill these resistant, these peptide-resistant breast cancer cells, where a peptide that works in a similar manner as NRCO3, a cationic peptide mastaparin, is not able to effectively kill these NRCO3 or NRC resistant cells. So we wanted to look to see whether or not these resistant cells are able to produce tumors effectively in mice. And in this paper here, we show that over injecting these resistant cancer cells, the NRCO3 or NRCO7 resistant cells, are not able to form tumors very well in immune deficient mice. So by looking at tumor volume and the mass and even the images of the tumors, they're not very good at producing tumors in mice. There are some potential effective ways to make HDPs even more effective. There are host defense peptides can be modified by adding a targeting peptide so that they will get to directly the cancer that you want them to get to. There are some caveats here. The linker may be susceptible to protease degradation, but it may mean that these peptides are more specific to a certain type of cancer or a certain type of cell. Another way to try to optimize these host defense peptides are to encapsulate them with a liposome, and then also to target that liposome specifically to a certain cancer cell, and so that they can get to the location so they can be more effective. So overall, the host defense peptides show some clinical promise based on some tissue culture and some preclinical studies. We still need more screening and more understanding to prove how selective they are and make them bioavailable so they're not degraded as easily in serum, but not killing our normal healthy cells. So this would be important so that we could have improved treatments for cancer. They may be useful in treating cancer stem cells to limit metastasis and cancer recurrence. Since these peptides are able to target the membrane, they may be good at targeting these cancer stem cells. They are potential solutions. There are potential solutions to make these peptides more effective, and with better understanding their mechanisms of action, we'll be able to better understand how they may be useful for potential drugs in future treatments. So we'd like to acknowledge and thank all of you for joining this session, both the Hoskin and Coombs labs and our collaborators, and these sources of funding. And I give you a couple images from Nova Scotia to show you where both of us are at Dalhousie and at Acadia University. So at this time, I'd like to introduce our next speaker. Dr. Hans Vogel completed his work and training in the Netherlands, which was followed by a PhD at the University of Alberta in Edmonton. He did a postdoctoral fellowship at the University of London, Sweden, and then joined the University of Calgary, and has over the years led an active and successful research group studying things such as calcium and iron binding proteins, metabolomics, and host defense peptides. And he is widely published and studies antimicrobial peptides, looking in areas of anti-biofilm, antifungal, and anti-cancer activities.
So thank you very much. Sorry for my very late arrival at the meeting. My internet at home crashed, and so I had to race to work and go into my office, a place where I haven't been for a long time, as many of you have been working from home.
Yes.
So thank you for the introduction, and thank you for the opportunity to present at this meeting. And I'm going to talk to you about some work that we have done on tryptophan and arginine-rich antimicrobial peptides. This has been a long-standing interest of my group, and today I'm going to talk to you about how these TRP and ARG-rich peptides can actually have anti-cancer activities. And in all the examples that I'll discuss today, I'll limit myself to studying the effects. I can also put on my laser pointer here. For studying the effects on Jurkat cell lines, which is of course a leukemia cell line that we have used for the majority of our studies, but we've also worked with a whole range of adherent cell lines. So why would one be interested in studying tryptophan and arginine-rich peptides? As we already heard from David, there are actually some peptides that you can obtain from protein in bovine milk. One of these peptides is called lactoferricin. And as I'll show you in a minute, this peptide is characterized by having quite a few tryptophans and arginines in its sequence. This is a peptide that's actually cut out from an intact protein. Now, there are a number of other antimicrobial peptides that have been found and characterized. For example, indolucidin is a bovine cathelicidin. Tritryptophan is one of our favorite peptides. It is a porcine cathelicidin. There are other tryptophan and arginine-rich peptides in wheat called puroindolin A and also puroindolin B. And in human lysozyme, there's also a sequence which contains, as you can see here, quite a bunch of arginines and tryptophans. And to top it all off, a large combinatorial hexapeptide screen identified only very few peptides that had a very good antimicrobial activity. And those peptides all had several arginines and several tryptophans in the sequence. So clearly, there is something special about the chemistry of the TRP and ARG-rich peptides that allows them to be a very good antimicrobial peptides. And this intrigued us, and this is why we started studying them. So this is the peptide that is really cut out of bovine lactoferrin. So this is in bovine milk. This is a protein that's very important. And then residue 17 to 41 get cut out. And as you can see, there's actually a region here that is very rich in arginines. There's 3 arginines here and 2 tryptophans. And this is the smallest sequence of this whole peptide that has been shown to have antimicrobial activity. Now, you may wonder why this occurs in bovine lactoferrin. This has also happened in human lactoferrin. So when you look at human lactoferrin, actually what is cut out is a much larger peptide. We now know that it looks like this most of the time. But originally people thought it looked like this, where there was a further cut made here. But that's been hard to reproduce in various other labs. Now, if you look in here, there's a tryptophan here, and there's a tryptophan here. They're very far apart from each other. But what is intriguing about this sequence is that there are all these arginines that are actually present here. And it turns out people have done a lot of studies on this 11 residue peptide, and that actually has cell penetrating properties. So we have made a series of peptides in which we have linked these as a cell penetrating peptide in front of some of these and variants of that in order to study them as anti-cancer peptides and more recently also as anti-biofilm peptides. Now, another interesting thing is that in the intact protein, this lactoferricin region is originally folded something like this with an alpha helix and a beta strand and then packed against the rest of the protein. But when you cut it out, it becomes this beta hairpin type structure. And you see it's now forms two beta strands together, form a small beta sheet with a loop, and it's held together by disulfide linkage here that you saw in the previous diagram. And this structure is completely amphipathic because all the positive charges are on one side of this beta sheet, whereas all the other hydrophobic residues are on the other one. And here you indeed see the two tryptophans as we found them to be positioned in the NMR structure. So this is a characteristic feature of many antimicrobial peptides, that they can form this amphipathic structure where you have hydrophobes on one side and positive charges in another region, and that seems to be necessary. And in subsequent studies by John Svensson and his group from the University of Tromsø, they made linear versions of this peptide, and they did an alanine scan throughout this 15 residue peptides. And as you can see, the two tryptophans are absolutely essential to that activity for this particular peptide. Any of the other residues can be replaced by an alanine with relatively minor effects on the antimicrobial activity, but the two tryptophans are really important and essential for that activity. And I have been trained as a chemist, and so this is intriguing to me. Why would this be specifically these tryptophans? And so we started to look into this. So this is the sequence that you find in lactoferricin, a bunch of arginines and tryptophans. And then if you looked at the combinatorial peptides that were uncovered, you see that they have a similar motif, a bunch of arginines and tryptophans. And then in human lysozyme, there's a sequence that occurs like that as well. And then indolicidin, a well-studied bovine cathelicidin antimicrobial peptide, you see something similar. In that case, there's even 5 tryptophans and again, positive charges, arginines and lysines. Our favorite peptide is tritrypsin because it has already a www, World Wide Web, right in the middle for the 3 tryptophans. And again, there's arginines very close by, and the puroinolines have similar motifs. So this motif clearly occurs many, many times. And most of these peptides, all of these indicated by this big arrow, and that one, are very active. This one's not so active because the 2 tryptophans are actually very far away from each other. And so it seems that you need to have the tryptophans quite close to each other. This one at the bottom here is not antimicrobial, at least not in our hands, because it just doesn't have enough positive charges. Now, one other reason that we were interested in this, we really like these peptides because they're shorter. If you look here, these are the peptides that I just mentioned, some of them to you. The combinatorial peptides are 6 residues short, and some of these other naturally occurring ones are 13 residues, whereas typical other ones like meguinin and LL37 are much longer, 23 residues or 37 residues. So that makes them much cheaper to synthesize. And we have done a substantial amount of NMR work on these peptides over the years, and they form very well-defined 2-turn structures when they bind to membrane mimetics. And as was already mentioned by Melanie earlier as well, this has led the company Lytix Biopharma in Norway to develop various peptides, and one oncolytic peptide is LTX-315, which was just mentioned in some previous slides. Now, why are these tryptophans so important, and why do we think they're so uniquely suited for this role? So the main reason that we think is that if you look at membrane proteins, the tryptophans are always found in the membrane interface. This is the hydrophobic core of the membrane proteins, and the hydrophobic core of these proteins always sits in the membrane interior. But the tryptophans in all the crystal structures that have been solved, you look at the red ones, those are the tryptophans. The orange ones are the tyrosines, they do the same thing. They always sit in this interface between the soluble phase and the membrane, interior of the membrane. And so this is probably what these peptides do. The tryptophans really help them to target to that membrane interface. But there's other reasons why tryptophans may be important. Tryptophan is quite hydrophobic, the indole ring, and so it likes to cluster various other hydrophobic amino acids around it when it's in water. And so this is what you can see, that you get these transient nucleation structures that form, and they may already help to give some structure that likes to interact with the membrane. And in addition, tryptophan can actually also stack with arginine residues, so that may be an explanation as why the combination tryptophan and arginine is found in most of these peptides. All right, so today's symposium is about anti-cancer peptides. So what is the relationship between antimicrobial peptides on the one hand and anti-cancer peptides on the other one? So several cationic and antimicrobial peptides have been shown to act as anti-cancer peptides. And we know that most antimicrobial peptides act by binding to negatively charged phosphatidylserine head groups of bacterial phosphatidylglycerol head groups of bacterial membranes. And these head groups are normally not found in human cells. They in fact have a neutral phosphatidylcholine head group. And so it's understandable that a positively charged, highly cationic antimicrobial peptide goes to negatively charged surfaces preferentially. Now, what happens in the cancer cell? In a cancer cell, the cell is no longer able to maintain the distribution of the phospholipid head groups across both leaflets. Normally, phosphatidylserine in a healthy cell would point out, but in cancer cells, a lot of them start to point to the outside of the cell, and that generates, again, negative charge on the surface of the cell, and that's why AMPs can act selectively on some cancer cells. And our trick is, of course, that we have to find those particular AMPs that selectively act, not the ones that kill everything, but the ones that selectively go after the cancer cells, i.e. the ones that recognize something. So when we started this research, we started reading the literature, and you can see there were two groups, one of them the Hoskin group in Halifax, that had already shown that this short peptide could not kill the cancer cells by itself, but it could do so when you inserted it in liposomes. And the Rekdal group, who is now the leader, Øystein Rekdal is now the leader of Lytix Biopharma in Norway, they had shown that this lactoferrin, this longer peptide, had anti-cancer activities, not only against cell lines, but also against transplanted tumors in mice. And they found that the disulfide bond was important for that particular activity. So we started our work in collaboration with Evan Haney and Bob Hancock in Vancouver, and later we transferred a lot of this program to Calgary. And we started to work with the leukemia cell line because it's relatively, the Urquhart leukemia cell line is relatively easy to work with because it is grown in suspension. And so we could screen a lot of peptides that we have in our libraries with these particular cells. And then we decided to use as controls not only red blood cells, which is what a lot of people have done in the case of antimicrobial peptides, but because this is leukemia, of course, occurs normally in the bloodstream, we decided to use PBMCs, which is a subset of all the white blood cells here, right? You can make this through centrifugation. So you separate the erythrocytes and the platelets on the other hand away, and then you're stuck with all these PBMCs. So we use those as control. And what we were looking for were, of course, for peptides that would kill a cancer cell, but would not kill the PBMCs, and certainly not kill the red blood cells. If you do that, then you kill just about everything. That we studied in one of our initial papers, which was published in 2017. And I'm not going to go over this except by saying that melatonin is actually a control peptide here. We know it's completely cytotoxic. And here are some of these hybrid peptides that I mentioned earlier, where we took the first part of human lactoferrin and connected it through various linkers with this active part of bovine lactoferrin. And here are the activities. And you can see that lactoferrin itself is quite active. These are done at 3 different concentrations. Melatonin kills everything. If you look here, melatonin also kills the PBMC. So melatonin is not particularly useful as an anti-cancer agent unless you can selectively deposit it into cells, but that's not what we're talking about here. Most of these other peptides were not as active. Some of these hybrids that we've constructed had some activity, but unfortunately, they also have a little bit of activity against PBMCs. So we don't think that they have a super bright future. So here's the red blood cells, and if you look closely at that, you see that melatonin even kills all the red blood cells under our experimental conditions, and that's the last thing you want to see in a peptide that has some selectivity. So when you calculate the ratio of killing uricate cells versus PBMCs, you see that lactoferricin, the 25 residue peptide, actually does quite well, as does one of the control peptides that we used, which is meguinin, which is a peptide obtained from frog. But most of the other peptides are pretty poor in being selective, and melatonin, of course, down here at the bottom, is totally unselective. So in a subsequent study, we started to play around with the arginines and the lysines in this particular group of peptides. And in this case, I'm going to talk to you only about this peptide tritryptophan that we've been working with, for which we have this available. And you can see that normally tritryptophan has 4 arginines in it, but we can replace those with 4 lysines, or with 4 ornithines, or with 4 diaminobutyric acids, or with an even shorter version of something that looks like an abbreviated lysine. You can use a longer arginine, you can use a shorter arginine side chain, and we also mucked around a bit with the tryptophanes by putting in a hydroxytryptophan, a sulfur-containing tryptophan, or we replaced the tryptophanes with tyrosines or phenylalanines. And then we started screening the activity. So this was a large group of 65 peptides that we screened, and this paper was just recently published. And what you can see in this particular case here is that if you look at the top diagram here and over here, you see the difference between tritryp arginine and tritryp lysine. And you can see that the lysine version is actually more selective, because the dark line is killing cancer cells, the other lines are killing the control cells. And you can see this actually, it gets even a little better if you put this abbreviated lysines in here. You see the activity, even at very low concentration, is much better, and the killing of red blood cells is almost non-existent. So these are actually very, very good analogs. And here are some results with some of the tryptophanes. We also mutated and removed some of the tryptophanes, but there was not much to be gained in terms of selectivity there. So then we screened this with flow cytometry, and you can see here by flow cytometry, to get a bit of an insight into how these peptides act. You can see, if you look here at the higher concentrations of 20 micromole, you see most of these peptides have a lytic effect on the cell. Etoposide, which induces apoptosis, doesn't do anything under the same conditions, but many of these peptides, they completely sort of have a lytic effect on these cells. The only one that's not particularly lytic is the one with lysine, but if you replace it with DAB, you can see that it's already almost completely a lytic peptide again. So we conclude that these peptides have a lytic mechanism, and we looked at the DAPI staining as well, and here etoposide gives the typical apoptotic bodies, but none of our other peptides give any of these apoptotic type bodies. And so again, this is consistent with the idea that we have a lytic mechanism for these tritryptophan based peptides. Okay, so I'm just going to remove this. Now, I just want to finish off by saying we've also looked at the tryptic stability of these peptides. And these peptides are very readily hydrolyzed by trypsin. You can see that here. It disappears after 1 hour, also when you replace it with lysine. But when you use the shorter or longer analogs of the of the arginine and the lysine, you see it's almost stable. So this is an added advantage of working with these abbreviated analogs, or even longer analogs of arginine, that in fact you now can make the peptides much stable. All right, so let me summarize all this, the final results. So what we've recently done then is we've shown that 3 of these naturally occurring antimicrobial peptides that are tryp and arginines rich are membrane lytic ACP. So that's a relatively new observation because that's not known. We've also shown that arginine into lysine residues, when you make that substitution, you get better selectivity, which is very important in this particular instance. Well, if you shorten the lysine into ornithine or DAB, diaminobutyric acid, you get improved ACP activity and therefore improved selectivity. I have not shown you any data, but we have a similar set of data for adherent prostate and bladder cancer cell lines, and we see similar effects in there. And finally, I showed you that ornithine and DAB peptides are much more resistant to tryptic digestion. So in summary, the conclusion of this last part of the talk is that manipulation of the cationic amino acids with unnatural amino acids such as ornithine. Ornithine is of course a natural amino acid, but it's not a protein amino acid. Diaminobutyric acid is unnatural. If you put those sorts of things in, or abbreviated or longer arginines, you can actually improve the selectivity and in some cases even the activity of anti-cancer peptides. And with that, I just want to thank the people who did all the work. Mauricio Arias in my group did a lot of the studies. Evan Haney did most of the studies with PBMCs and the control cells. And Ashley Hilsey, now in Halifax, did a lot of the work with the FACS and the DAPI and helped us also in other ways. And these are our collaborators across the world, and these are the funders of our research. And I thank you for your attention. And again, my apologies. This is not the setting in which I'd wanted to talk to you, but as I said, the internet at home crashed. I'm happy that I could be here and give my talk. Thank you.
Thank you. I'm happy that you were able to make it on. We do have one question so far in the question and answer. There's a Q&A, and the question is, would there happen to be an explanation for why tryptophan seems to be more significant in HDP action as opposed to the other aromatic amino acids?
Yeah, absolutely. That's a very good question. Let me see. I may even. Well, let me first try to address this directly. So tryptophan is, of all the amino acids, the indole ring of tryptophan is quite unique in the sense that it is the biggest aromatic ring system that there is amongst all the amino acids. And people have shown, and this is found in numerous crystal structures for memory proteins that have been solved, that tryptophan likes to sit in the interface of biological membranes. That's where it's always found. And so tryptophan-rich peptides would love to sit in the interface. And if you can perturb the interface and in that way maybe enter a membrane, you can perturb cells through a lytic mechanism, because as I showed you, tritryptophan is mostly a lytic peptide. These shorter peptides, the 6 residue peptides, they don't have a lytic mechanism. We think they actually transfer spontaneously through the membrane to the inside of the cell, but you have to encourage them to do that. And that's why we attach things like cell-penetrating peptides to them. Otherwise, they just stay stuck on the membrane and nothing much happens. So that's the main reason we think that tryptophans are unique. And why are they different from phenylalanines? If you look at the distribution of phenylalanines in membrane proteins, they're always right in the middle of the membrane. So they're in a completely different orientation than where the tryptophans are. The tyrosines behave similar to the tryptophans in that they like to sit in that interface, but they seem to be more hydrophilic, and they seem to have a harder time to go deeper into the membrane. We've recently published a paper about that where we used fluorine NMR to look at that in some detail. And the impression of that study was that tyrosine really prefers to stay on that membrane surface and not go into the interior of a membrane, whereas tryptophan can do that. Now, the other thing tryptophan can do is it can collect things around it while it's in solution. And so that may already help to get things targeted to the membrane. It can also form interactions, bi-stacking interactions with arginine, and that may help it to actually transfer through the membrane as well.
Great, thank you. There's a couple more questions that have come through.
Okay.
Is there any increase in hemolysis after the modification of cations and natural amino acids?
So if we go back a few slides, I don't know, can you still see the slides?
Yeah.
So the question was, is there any increase in hemolytic activity? So if you look here, you know, hemolytic activity here for ornithine is completely gone, basically. This is red blood cells, right? This is for DAB, right? Looks like I've lost my my laser pointer. You can also see there is no hemolytic activity really measured for these two peptides. And you compare that to the native tritryptophan or the one with the arginine, you see there is a substantial effect still at higher concentrations towards. So actually, we've got improved selectivity. You see that there's still some effect at the higher concentrations here on the white blood cells, the PBMCs.
Right.
More so here than there. But the good thing is, look at how active these peptides are at low concentrations compared to all the other ones, right? So if you look at low concentrations, you see that these things are already 100% active at very low concentrations. And at those points, there's almost no action on PBMCs and certainly no actions on RBCs. So these points, these 2 points in all these diagrams are the ones that make us excited, because that's where you have the improved activity and you have the excellent selectivity compared to all the other peptides.
Thank you. That does help clarify, I think, for the person asking the question, and it is exciting to see that happens. There's another question here. Is there any evidence of using AMPs and antibiotics in synergy to combat antibiotic resistance?
We have personally, we've never really looked at creating resistance. I know you just talked about it in the comments that you made earlier, but we do know for many antimicrobial peptides that when you do combined treatments between antimicrobial peptides and regular antibiotics, that there is a synergistic effect in many cases, not in all cases, but many authors have reported those sorts of synergistic effects. My impression is that if you have a peptide that is somewhat lytic, it will make perturbances in the membrane and perhaps make it easier for an antibiotic to get into the cell. But that has not been rigorously studied yet. There are other antimicrobial peptides that have a non-lytic mechanism. They interfere with things inside the cell. And I don't know if it's actually known if they combine very well with antibiotics, but that would be a very interesting question to study.
Yes, I think it would be interesting. There is one other question that just popped through. Do they have any effect of these AMPs on immune response?
That's a very good point, and I'm not the one to really address that. I think Evan Haney has done a lot of work in that area in Bob Hancock's lab. I don't know if it's in his presentation for today, but yes, many of these peptides can have effects on different immune cells, and we've just not rigorously studied it for our groups of peptides. So I'd rather not, make comments about it, because I'm not that well informed on that topic.
Great, thank you. I appreciate that. I don't see any other questions coming through. I thank you for sharing, and I'm glad that you made it to be able to present today for the webinar.
Thank you.
So our next speaker, I'd like to introduce Dr. Valerie Booth. Valerie became a biophysicist. She enjoys working between biochemistry and physics. She holds a Bachelor of Science in Physics from University of Victoria. She did her MSc in biophysics at the University of Waterloo, and she has a PhD in medical biophysics from the University of Toronto. She's been a professor at Memorial University in Newfoundland, and she's researching lung surfactants, antimicrobial peptides, and molecular crabbing. So I'd like to welcome Dr. Valerie Booth to share with us a little bit about the potential of her research in antimicrobial peptides.
I'd like to thank Dr. Coombs and Dr. Hoskin for organizing this meeting and for asking me to speak here, as well as all of our audience members who've shown up to listen. I thought I would take a second to explain a little bit of the story behind the picture on my title slide, because I think injecting a little bit of humanity into these virtual events is pretty important. So this picture is from a couple days ago when I was on a family backpacking trip on the East Coast Trail, which is a 330 something or other kilometer trail that goes along the east coast of Newfoundland near my house in St. John's. We took 5 days and we did like a quarter of the distance of the whole thing. So I think we had to come back for another 3 years and try to finish it off. But in the meantime, my body's now stiff and sore, so it's a good time for sciencing. So I have divided my talk today up into two little mini themes. So first off, I'm going to talk to you about our studies of two histidine-rich host defense peptides that are of particular interest for potential use against cancer because of that reduced pH that tends to be around solid tumors. And for this part, I'm going to focus on detailed mechanisms, especially of interactions between the HDPs and membranes as studied in model systems, so just simple mixtures of lipids and peptides. Then I'm going to switch gears a little and I'm going to talk about how we can do the same kind of studies, but moving into more complicated systems like whole bacteria, and especially what happens to those membrane interactions when we start messing with the non-lipid parts of the bacterial cell envelopes. And I'm going to try to make some connections between that work in bacteria and what I think it has to teach us about how to optimize the interactions between HDPs and cancer cells. So the two peptides that I'm going to talk about are from cod, which is an important organism for Newfoundlanders. It used to be economically important. Now it's just kind of culturally important. And we have focused on two peptides from paralogous genes in cod, which we call gadacidin-1 and gadacidin-2. And as you can see in the yellow highlighting in the sequences, they're both rich in histidine residues. The table that's there at your bottom right shows you the expected charge on each peptide at neutral pH, so pH 7 versus a lower pH. So at a lower pH, we're expecting these peptides are both going to be more positively charged, and kind of a simple assumption from that would be like, oh, if they're more positively charged, maybe they'll be more active. And it turns out that this simple assumption turns out to be reasonably true for gadacidin-2, but in general not true for gadacidin-1, which is something I'll circle back to a bit in a few slides. In general, in terms of function, GAD1 tends to show similar activity at pH 5 compared to pH 7. GAD2 tends to show greater activity at low pH compared to neutral pH. And of course, the nuance is exactly, which cells you're testing it against in these studies. Also, GAD1 is more active than GAD2 at both neutral and lower pHs. And these functional assays are both from minimal inhibitory concentration assays that we do in my lab, as well as, for example, the myeloma cell assays done with collaborators, Melanie R and David Hoskin. So, okay, so I'm going to just quickly cherry pick a few results from our model system studies of these gadacidin peptides. So we have experimental structural data that shows that the level of helicity is very dependent on the lipid composition of the membrane, and some structural plasticity of the peptides is at play, especially for gadacidin-2. We've got computational data, which is what is shown here, which shows how individual gadacidin peptides are able to make a defect in a membrane all by themselves. And the kind of interesting thing that came out of the molecular dynamic simulation was that there's something special about histidine pairs. So not so much histidines by themselves, but when you have one histidine right after another in the sequence, they have a really high propensity to locate right at that polar-apolar interface in the membrane. And it doesn't seem to matter if they're charged or not, okay? Which is a little bit of a hit to our initial assumption that more charge equals more activity. We've done some binding studies in collaboration with Dr. Mike Morrow using zeta potential measurements to measure binding to liposomes made out of POPC and POPG. And this work was motivated in large part in trying to better understand the reason for the greater activity of GAD2 at low pH compared to neutral pH. Turns out that binding to POPC, POPG follows nicely with GAD1's greater activity compared to GAD2, because you see tighter binding for GAD1 compared to GAD2. But it doesn't actually help us understand the story of GAD2 and pH, because it binds pretty weakly at both pHs. Although we don't see a big difference in membrane binding for GAD2 with altered pH, it's still possible that there's a difference in how much GAD2 disrupts membranes at the two pHs. So maybe there's more membrane disruption at low pH for GAD2. So that's the next kind of study I'm going to show you, which is deuterium to study how it is that the peptides are disrupting the membranes. So I'm going to spend a little bit of time here because I want to explain this technique for people who aren't familiar with it. It's a technique that we use to study what the HTPs are doing to the lipid bilayers here in model lipid systems, but in a few minutes I'm going to show you the same thing, but in the context not of model membranes, but of actual whole live intact cells. So the first thing to know is that since we're using deuterium NMR, we do need to use a sample that has deuterated lipid acyl chains. The next thing, if you just look at the spectrum, is just to notice that it has this beautiful kind of bat-wing shape, because each deuteron in our deuterated lipid chain is giving rise to a pair of peaks. And the methyl peaks, which are the ones near the center of the spectrum, shown by the little short blue arrow, those are the ones with the narrowest splittings. The deuterons near the head groups give the widest splittings, so the longer blue arrow. And really, the number one thing to remember about all these spectra is that smaller splittings indicate more membrane disorder. Okay, so that's why the methyls have the narrowest splitting, because they're in the middle of the bilayer where things are their most disordered. And we're going to use this property. Okay, how we're going to use it is when we add our HTP, if it disrupts the membrane, we're going to expect to see all of the splittings get a bit narrower. Okay, so we're going to see the membrane get more disordered. So let's have a look at this with GAD1 and GAD2, which are studies again done in collaboration with Dr. Mike Morrow. And so here's our same kind of bat wing spectral shape. They look a little bit squashed because I made space on top of them to put the data with the peptides. So in gray and in white, we can see lipids by themselves at the two pHs, so no change in membrane order at the two pHs. If we add in gadacide 1, we see, as expected, the splittings get narrower, meaning that the peptide has disrupted the lipids. So one thing to note is that there's no more reduction in membrane order at pH 5 compared to pH 7. The splittings are pretty much the same, which goes with the insensitivity of GAD1 function to pH change. GAD1, doesn't really care what the pH is. The story for GAD2, which is the peptide that had the pH-dependent activity, it's more active at low pH, is similar in some ways to what we see for GAD1, because we see a reduction in splittings, which tells us that the peptide disrupts the model membrane. However, with GAD2, there's not quite so much membrane disruption as for GAD1. So not quite such a big reduction in splittings with GAD2 as compared to GAD1, which fits with its activity because it's less active than GAD1 at both pHs. So the thing that does not fit with GAD2's activity is that there's no more disruption at pH 5 than at pH 7. Okay, so GAD2 is more functional at pH 5 than pH 7, but we don't see that reflected in the membrane disruption in these POPC, POPG vesicles. So that gives a little bit of head scratching, but there's a whole bunch of possible explanations for why membrane disruption doesn't explain why GAD2 is more active at low pH. And a lot of those potential explanations come into the category of, things that are probably no big surprise to any of the people here, which is that there are a lot more things for HDPs to interact with in cells besides just the lipids. So this moves us towards the second half of the talk. So let's have a cartoony look at cells and what might be important in interactions with HDPs besides the lipids. And we can split these non-lipid things up into things in the cell envelope, like for bacteria, peptidoglycan, lipopolysaccharide, teichoic acids, as well as things that are inside the cells, of which there's too many that I didn't even feel I wanted to attempt to draw them. And I will show you in a few slides one example of an intracellular target. And of course, at the same time, we're thinking ahead to eukaryotic systems and thinking about interactions with things, you know, on cells like heparin sulfate that might be potentially important in interactions between HDPs and cells. Okay, so here is what I see when I look at this problem. On the one hand, there is the world of HDP function. For example, these minimal inhibitory concentration assays to figure out how much peptide you need to inhibit growth. And on the other hand, there's the world of these kinds of molecular studies of HDP mechanisms of interacting with lipids. So like the studies I've shown you with NMR, but also a whole pile of other types of experiments, including things like dye release assays from liposomes and so on. And there's one way to appreciate how really, really different these two worlds are, and that is to compare the ratio of peptide to lipid in the two types of studies, because it turns out they are crazy, crazy different. To see growth inhibition, we're looking at an HDP to lipid ratio of about 100 to 1. Okay, so that's in a standard MIC experiment. So lots of peptide compared to lipids. In our model system studies, we actually only need something on the order of about 1 peptide per every 100 lipid molecules to start to see disruption. Okay, so 100 to 1 peptide to lipid ratio versus 1 to 100. So that gives us a difference of 100,000 times in peptide to lipid ratio. So I am very interested in how we can bridge this gap. Okay, so how do we bring these two worlds together? We've done this in a few ways, and the one way that I'm going to focus on here is, of course, deuterium NMR, which I got you all set for just a little while ago. Okay, so remember a few slides ago, here's a beautiful batwing spectrum that we get, and we can see that it gets narrower when we add HDP and we have disrupted lipid acyl chains. So we spent some time developing a way to do the exact same experiments in membrane lipid deuterated bacteria in place of the model lipids. Okay, so I'm just slapping some bacteria on, and I'm changing our title from POBC, POBG to deuterated bacteria. So same experiment, all that changes is now that instead of our lipids being all by themselves, they're actually in bacteria. So how do the spectra of these membrane deuterated bacteria look? You can see the model lipid spectrum in white for reference, and the spectrum from our membrane deuterated E. coli and Bacillus subtilis in the colors. And here you can see that something pretty terrible has happened to our bat wing shape. Looks like our bat had a pretty rough night out. The bacteria, however, they're actually in pretty decent shape. We can put them in the NMR spectrometer alive, leave them there for 8 hours at 37 degrees, and at the end, at least about 80% of them are still alive when we take them out at the end of the experiment. So if we turn to actually looking at these ugly looking spectrum from the bacteria, they can be ugly, but they're still rich in information about what the lipids in the bacteria are doing. For instance, you can see that we're quite interested in the splitting at the prominent edge, which I've got marked in the dotted lines, that comes from the part of the lipids near the head groups, and that it stays the same regardless of whether the lipids are by themselves or in bacteria. And as I summarized on this slide, we've played around quite a lot with these kinds of experiments. And I have to warn you though that I don't have spectra to show you with the GATA side and peptides, because these whole bacteria experiments burn through a ton of peptide. We, you know, back in the good old days when we did model membrane studies, we used to order peptide in, you know, tens of milligrams, and now we've had to start ordering it in, you know, 5-gram batches instead. And so we have picked on peptides that synthesize well and have been well studied by other methods. So many people will be familiar with some of the HDPs that we have looked at, because they include things like MSI-78, BP100, and CAME. And we've looked at both gram-positive and gram-negative bacteria now. And what we've been playing with most recently is characterizing how the AMP lipid interactions change if we start messing around with the non-lipid components of the cell envelope. Okay, so far we have looked at lipopolysaccharide and peptidoglycan. So let me take you through some of this work. Let's start by looking at the spectra on the right. So these are spectra of membrane deuterated E. coli as we add progressively more of the HDP MSI-78. So more as you move from the bottom of the spectra towards the top. And the percentage gives the percentage weight of peptide per dry weight of bacteria. And we can see that as we add peptide, we lose intensity at those prominent edges indicated by the arrows, and we gain intensity near the center of the spectra. So we can see the same kind of lipid disruption in whole bacteria as we do in the model lipid studies, which for me is still kind of a, wow, cool result. The other thing that really strikes me about this work is thinking again about those peptide to lipid ratios. So if you go into the math, what I've got shown as 30% MSI-78 works out to be roughly a protein to lipid ratio, mole ratio of about 1:1. Okay, so remember the two worlds that we're trying to bring together, which were the HDP function realm, where we need about 100 peptides to each lipid to see growth in inhibition. And on the other hand, the model membrane system studies of HDP mechanisms, where we need about 10,000 times less peptide for every lipid to see membrane disruption. With this 1:1 mole ratio, we did manage to split the difference in between the two. So that's kind of cool how that came out. So now, given that we have to add 100 times more peptide to see the same lipid disruption in our intact bacteria as in the exact same experiment in lipid-only system, it kind of begs the question, you know, what are those other 99 peptides really doing in the bacteria? And to split it up in a really simple way, we can think about, well, maybe the peptide is sticking to something outside the cell, or maybe it's sticking to something inside the cell. And number two is, you know, suggested in large part by, these are actually pretty big membrane disruptions. So it's hard to believe that with this much membrane disruption, our peptide isn't getting inside the cell. Okay, so the first one I'm going to take on is a story about what might MSI-78 be sticking to inside the cell, because I can make it a quick story. We started looking for the intracellular target in kind of a funny way, and that was with a technique called differential scanning calorimetry, because this is part of the theme of Valerie challenging students to put complicated samples into instruments that are traditionally used for much simpler systems. So what I was actually hoping to see was MSI-78 maybe interacting with the carbohydrate components of the cell envelope. And that's not what we got, but what we got was actually really striking, because it's, you know, if you can see the data, it's a bit shocking. So MSI-78 just nails the ribosomes, so totally destabilizes them thermodynamically. And then we went back and we confirmed that with a more conventional approach, which is a transcription translation assay. And MSI-78 is pretty well studied, but I have not actually seen another reference to MSI-78 targeting ribosomes, but certainly in our hands, we find that it does. Okay, so back to thinking about what the rest of the MSI-78 is doing. Okay, if we need so much more peptide to induce lipid membrane disruption in bacteria compared to just model membranes, Now we're going to think about what are the non-lipid things in the cell envelope that MSI-78 might be sticking to. All right, and there's kind of two ideas floating around for how non-lipid components of bacteria might modify HDP lipid interactions. And of course, we're always thinking ahead to how the non-lipid interaction, how the non-lipid components of cancer cell envelopes might matter as well. So number one, non-lipid cell envelope components may act as kind of magnets, attracting more peptide near the membrane where it can do damage. Or number two, non-lipid cell envelope components might act as sponges, kind of soaking up the HDPs and preventing them from reaching the membrane, so kind of protecting the cells. So the rest of what I'm going to show you is our quest to kind of look at these possibilities for lipopolysaccharide and for peptidoglycan. So I'll start with lipopolysaccharide, and before I get back to NMR, I wanted to show you one shot of molecular dynamics simulation data that a grad student in our group, Shayla Montero, is carrying out right now. She's working on coarse-grained simulations of the outer membrane of E. coli with all the lovely carbohydrates sticking out, shown in red and brown sticks near the top of her snapshot. And her peptide in the middle, you can just barely see, is kind of nestled in there in blue. And she's found something interesting, which is there's a particular spot in the inner core of the carbohydrates where the peptide just loves to sit. And it's actually hard to move the peptide out of that spot. Okay, so back to NMR experiments. Another current student, who's Sarika Kumari, she has been gently disrupting her E. coli with an EDTA treatment to destabilize the lipopolysaccharide without killing the cells, and then asking if that LPS disruption enhances or inhibits her HDP-induced membrane disruption. So first she had to show that her EDTA treatment doesn't kill the bacteria or disrupt the NMR spectrum of the membrane deuterated E. coli, and that's what's shown in white in this figure. Next, she did a MSI-78 only treatment in blue, which shows the usual change in spectral shape that signals there's membrane disruption. And then she did the combination where she both destabilized the LPS and she added MSI-78. And she found that the EDTA disruption of the LPS allows the peptide to disrupt the acyl chain somewhat more, and that's shown in red. And importantly, she's done this more than once, so we think it's a robust result. Okay, the last data I'm going to show is the peptidoglycan part of the story. And this is work from a recently graduated PhD student. So now she's Dr. Nuri Santisteban. And for these, she used gram-positive bacteria. She used the gentle lysozyme treatment to disrupt the peptidoglycan without killing the cells. So if you've ever made spheroplasts, that's basically the technique she uses. In this case, she found that just disrupting the peptidoglycan slightly increased the lipid disorder, even in the absence of HDP. For the combo treatment, where she disrupted the peptidoglycan and also added peptide, she didn't really see any extra effect over peptide alone, at least within the normal sample-to-sample variation in the spectra. And now, of course, I'm really interested to know what happens if we do something to the teichoic acids, because they have a negative charge instead of PGN. But I haven't really started thinking about how we're going to do that yet. Okay, to sum up, on the histidine-rich GAD-aside story, we can say that model system experiments do correlate with greater GAD1 activity over GAD2 activity. Model system experiments don't correlate well with GAD2 pH-dependent activity. From the intact cell NMR, we've learned that the HDP to lipid ratio needed to see lipid disruption in intact cells is about 100 times more than needed to see the same disruption in model systems. Disrupting LPS increases HDP membrane disruption activity, at least for the one HDP explored thus far. Disrupting peptidoglycan of gram-positive cells has little effect on HDP membrane disruption activity. And what this kind of work has emphasized for me is that we should be optimizing HDPs not just for membrane disrupting activity, but also for non-lipid cell envelope components and beyond. Okay, and I'm sure I'm not the only person who's thought of this, so that's why I've said emphasized. So for the future, now that we have tools to probe molecular mechanisms of HDP lipid interactions in both model systems and intact bacteria, I want to know if we can extend the whole cell techniques to eukaryotic cells or organelles, maybe something like mitochondria. All right, so the sort of drawback that we have with this work is it takes a ton of HDP, so it's not something we can do without, at least a gram of it. And a drawback for us is just that we've never grown anything more complicated than bacteria. So now we have to figure out how to get deuterium NMR labels into eukaryotic cell membranes. So with that, I would like to thank the people who did the work, many of whom are shown here. And this is in our last group picture, kind of in the before times when we were actually all together. And I hope I've properly credited everybody on my way through the presentation. And I'd like to thank you for your attention.
Great, thank you, Dr. Booth, for sharing some of your research with us. One question that I had is, have you looked at any or made any model systems that might be closer to cancer cells? You mentioned eukaryotic cells are something that you'd like to look at. Have you thought about how to make one?
We haven't done it in model system studies. Kind of the one step towards like real cells that we've taken is we have done things like add cardiolipin, but that's more thinking about bacteria. But yeah, that is the thing that we could do, is pick lipids that just better match what we would find in a cancer cell.
Cool. There is another question here. How much is it likely that molecular crowding could be the reason for impairment of many cellular functions, leading to major diseases like cancer, cardiovascular diseases, diabetes, and Alzheimer's disease?
I would say big, but I'm not sure that I'm the right person to answer the question. So I think it's a really interesting question. I'm very interested in crowding, and yeah, I would be interested in what somebody who is an actual expert in that field would have to say.
Thank you very much for participating in our webinar and helping us kind of see what some of the NMR looks like and explaining it. We were looking in my lab recently at some of those bat wing shaped and trying to understand them, and I really appreciate you talking us through them.
Oh, now you know like the people who to talk to try to like, yeah, figure those things out.
Thank you very much. I really appreciate your participating today. Next, I'd like to welcome our next speaker, Dr. Evan Haney. He graduated with his Bachelor in Science in Biochemistry and got a doctoral degree both from University of Calgary in Canada. He did his PhD work with Dr. Hans Vogel, who we just heard from a few moments ago. And he was characterizing antimicrobial peptides, looking at antibacterial mechanism of action. He's currently a research associate in the laboratory of Dr. Robert Hancock at University of British Columbia. And he's looking at the host defense peptides and their potential as therapeutics, looking at antimicrobial effects as along with immunomodulatory, antibiofilm, and anti-tumorigenic properties. So thank you very much, Dr. Haney, for participating today, and interesting to hear what you have to share.
All right, thank you very much. Hopefully my slides look correct and everybody can hear me. So let's go ahead and get started. Thank you so much for the opportunity to come and share with you some of our work that we've been doing looking at host defense peptides in cancer. So we saw some pictures from the East Coast. I'm all the way over on the West Coast of Canada. So here's a picture of UBC. And as mentioned, I work in Bob Hancock's group here at UBC, and we're very interested in host defense peptides. Usually I live sort of on this side of this diagram. So we know that host defense peptides have a broad spectrum of activity. Everybody's very excited. And it's always talked about the therapeutic potential of these peptides. So we're very interested in looking at them in terms of infectious diseases. So we've looked a lot at their anti-biofilm activity and their direct antimicrobial activity. We've also been involved heavily in looking at their immunomodulatory activity, so their effects on the immune system. Today, I'm going to do a little bit of a departure, something that I only dabble in a little bit, is looking at their anti-cancer effects and then also looking at their cytotoxic effects. So we've already had a great introduction. We're all very familiar with this today from everybody's talks already. But just really quickly, anti-cancer peptides, I would consider them a subset of host defense peptides. They're usually short peptides, and as we've seen, they're often amphipathic in some of their structures, and they often carry a cationic charge. They're typically broad spectrum in terms of cancer cells. That's usually meaning that they're able to kill lots of different types of cancer. And we think of them in terms of two distinct mechanisms of action. So the first one we call direct acting or lytic, where they actually essentially blow up the cancer cells. And the other one would be indirect acting, where they would induce apoptosis and cell death pathways. The nice thing for cancer is they don't target cells based on growth rate. So if you compare it to traditional chemotherapy that often target dividing cells, anti-cancer peptides aren't really using that kind of mechanism. And something that I'm not going to get into too much, but it's kind of been hinted at, is kind of this immune response. Be happy to discuss some of it. But they are capable of inducing an anti-tumor response, although I think that the peptides that may induce immune response actually might be sequence specific or distinct from those that are killing cancer cells. So anti-cancer peptides have been identified in a lot of places in nature. So that's just looking at from some of the classic ones from silk moss, so sacropins. We've heard talk of lactoferricin peptides, as well as melittin, which is a bee venom peptide. So they're found all over the place. Really quickly looking at their mechanism of action. So I mentioned this quickly. If you look at the difference between a lytic peptide versus an apoptotic inducing peptide, the lytic peptides will obviously, when you treat cancer cells with them, they're going to have a huge effect on the membrane structure, and you'll get large disruption at the surface of the membrane. If you look at the effects of an indirect acting peptide, or one that's apoptotic, you won't see those same effects on the membrane surface, of the outer surface of the membrane. But for the purposes of today's talk, you'll be seeing some data where we're looking at direct acting features of peptides. And so that'll include things like altered cellular morphology. We'll be looking at propydium iodide uptake assays, as well as just looking at membrane damage at the surface of the cells. If we're talking about indirect acting features, cells that that cause peptides that cause apoptosis, you'd be able to see things like DNA laddering upon peptide treatment, chromatin condensation or nuclear blebbing, and then also in most cases, if you block caspase pathways, you can actually then block the killing. So that would be kind of how we look at it. So it's just something to keep in mind as we look at some of the data that I'm going to present today. So I've broken this up into two parts. They're very short. The first one is going to be the identification of a host defense or anti-cancer peptide through peptide library screening. And then the second part will be looking at strategies that we've used to try and optimize a peptide to improve its selectivity for cancer cells. So this first part, how do we identify peptides with anti-cancer activity? Well, working in a peptide lab, and I'm sure Dr. Booth and Dr. Vogel can attest to this. After you do this for several years, you end up with freezers full of peptides. And so several years ago now, we got into doing some of this work with Dr. Hilchi, who's been mentioned a couple of times today. And she came to Bob's lab and wanted to look for anti-cancer peptides. So we said, well, let's go to our freezer and we'll pull out a bunch of peptides and let's just screen and start looking at it. So we selected a subset of peptides from our freezer. And we screened them for activity, cytotoxic activity against 2 breast cancer cell lines. So the MDA, MB436 and 231 cell lines. And so using an MTT assay, we just wanted to see peptides that could cause substantial death in both cell lines and that were ideally cytotoxic at low peptide concentrations. So if we look at the overall data here, we find that a lot of these peptides do have effects on these cancer cell lines. So you can see that at fairly high concentrations, a lot of the peptides that we took out of the freezer were able to kill those cancer cell lines. But 3 peptides that I'm going to highlight in particular, this V681, mastaperin MO, which means modified, or mastaperin, you can see that even at low peptide concentrations, we're having a large effect on the cancer cell lines. So these are peptides that we decided to move forward with. I'll point out a couple other peptides that have been mentioned today. So the NRC03 and 07 peptides that Dr. Hoskin mentioned. We included those in our panel as sort of a control to make sure they also showed up in ours, so that was good. But the ones that we focused on were these, and what we did was a hemolysis counter screen to rule out just a general toxin. So we took those same 3 peptides and we checked them for their hemolytic activity. In this case, V681 and mastaperin, this modified form, were very hemolytic under our conditions, but mastaperin wasn't hemolytic at any of the concentrations that we tested. So this was the peptide that we decided to move forward with and look at a little bit more in depth. So just for those of you who are unfamiliar with mastaperins, it is a peptide toxin from wasp venom, so it's similar to melittin in that case. The peptide that we're looking at is 14 residues long and has a +4 charge. The sequences of it is here. You'll see it has positively charged residues of lysine, so there's 3 lysines in the sequence. There's no tryptophans in this peptide, but it's rich in leucines and alanines, and so this peptide actually, you would expect it to adopt a fairly helical conformation, particularly in the presence of membranes. Early studies found that this peptide is mediated mast cell degranulation and histamine release, so it's one of the reasons that you get swelling when you get stung by a wasp. And more recent work has found that it has anti-parasite activity and also has antibacterial activity. But what we were interested in is looking at its anti-cancer effects. So based on the results of our first screen, we started screening mastaperin against pretty much any cancer cell line we could get our hands on. And we found that it quite readily killed both hematopoietic and non-hematopoietic cancer cells. So we're able to get almost complete killing at 25 micromolar and 50 micromolar of several blood cancer cell lines and brain cancer cell lines. Several breast cancer cell lines were also affected in a concentration-dependent manner. And even chemotherapy-resistant cancer cell lines were also affected by the anti-cancer effects of mastaperin. So that was really exciting for us. We went on to characterize the mechanism of action for it, and we found that mastaperin is a direct-acting anti-cancer peptide. So you've seen some of these before, but this is the PI uptake assay. So if you treat Jurkat cells with mastaperin, you see an increase in PI staining compared to untreated cells. And so that would indicate that the cell is damaging the membrane. The PI is, the propionate iodide is able to go inside the cell. We ruled out the apoptotic mechanism of action by looking at DNA banding of Jurkat treated cells. So here you can see that if you use etoposide, they're faint there, but you do see banding pattern that would be characteristic of your apoptosis pathway. But the peptide treated cells, they really only resulted in DNA smearing. So we think that this is indicating that the peptide is just blowing apart the cells. And again, we see that too if you look at DAPI staining, where peptide treated cells, you start to see the cells blow apart and the contents get released. Whereas if you look again at the etoposide, you see the presence of the apoptotic bodies that would be more characteristic of an indirect peptide. We also went on to characterize this in vivo. So we used a mouse model of breast cancer, and in this case, we took 4T1 cells and injected them in the fat pads of BALB/c mice. And we treated them with gemcitabine, mastoperin, or the combo of the two. And you can see here now that treatment with mastoperin reduced tumor volume over the course of the experiment. And actually, the combination of the two worked to almost completely inhibit tumor growth, or very drastically reduce tumor growth. At the end of the experiment, you can also see that actually the combination worked the best in significantly reducing tumor mass, and you can see that down here, just some representative tumors as well. So this peptide worked great. We were very excited about it, and just getting ready to put our paper together. And this paper got published, and they found that mastoperin actually acted as an indirect anti-cancer peptide, which obviously didn't line up with what we were finding. And in this paper, what they actually used was an unamidated form, or a free carboxyl group of the peptide. And so in our case, we were using the amidated form of the peptide, and so we made the free carboxyl form. What we found that even just this subtle change in the sequence, or in the structure of the peptide, actually had a huge effect on the activity profile and what it could do. So you can see here that the amidated form of the peptide was highly active against Jurkat cells, whereas the free carboxyl form, you really didn't see that effect as strong as the unamidated form. Now I've included this here, maybe partially because my PhD supervisor is on the call, and so I just wanted to show him that I could still do the stuff that I learned in his lab. But what we ended up finding was that these peptides, this amidation actually contributed a lot to its ability to interact with vesicles and adopt a helical conformation. So if we look here, these are vesicle leakage studies where we find that and what I've got here is shown. We've gone from a POPS, which would be a negatively charged membrane, and then we bring it up to less negative charge, more zwitterionic, so POPC. We do increasing concentrations of POPS here. What you can see is as you, I guess I'll speak to it as you decrease the negative charge, you can see that for mastoperin, you actually don't have a huge effect on its ability to disrupt the membranes. Whereas if you look at the free carboxyl, you can see now that as you decrease the negative charge, the amount of release that you get goes down. And so this would indicate that the peptide is not disrupting the membrane as much. When we look at in terms of folding, we found that both peptides adopted helical conformation in the presence of various micelles and lipid environments. But if we looked at their ability, like the propensity of the peptide to form a helix, we did this with the TFE titration, we find that the mastoperin more readily adopts a helical conformation than the carboxyl group. So this would suggest that the amidation causes greater vesicle leakage, more readily adopts a helical conformation. We think that's why it's more effective as an anti-cancer peptide. So now that we've identified a peptide as being good, as having good anti-cancer activity, we want to go about making a better peptide. That's what we try and do. And so the sort of the classical way of doing this is just doing rational design, and people have done this a lot in the literature, where we could guess at important residues in the mastoperin sequence and generate rational variants. You know, we might mess around with some of the positively charged residues. We might introduce a few tryptophans here or there, and we could maybe do a small peptide library and see what works. But really, we would be guessing at it, and anything that we would get out would be probably luck. The philosophy that we like to take in the Hancock lab is to use more of an activity-guided design approach, where we want to screen as many peptides as we can, and then use the activity of the resulting activity screen as our to drive the selection of our new peptides. So in this case, we wouldn't be biased to any specific physicochemical property. We wouldn't be kind of focusing in on specific residues. We're really trying to get a broader view of what's going on. Now, the activity guided design requires that we have a huge number of peptides. And as has already kind of been alluded to by some of the other speakers, peptides are pretty expensive. And if you know, you want to start making hundreds of peptides, you could go broke pretty quick. So the technique that we use to get around that is peptide arrays, or spot synthesized peptide arrays. And so this is a technique that's been around for 15, 20 years now. But what it lets you do is you can actually make synthetic peptides on a cellulose membrane support, and just using standard synthetic peptide reactions, you can add on at each one of these different spots the amino acids of interest, and it allows you to build up peptide sequences of a defined sequence, but it gives you a much smaller amount, but you can do it at a much more reasonable cost. So here, for instance, would be an example of a spot array, where each one of these spots would be a separate peptide sequence. We actually get these made commercially, so when we order them, they get delivered to us in these microfuge tubes. I think this might actually be the anti-cancer peptide library that I'm going to talk to you about. But this whole technique allows us to obtain large numbers of sequences at a fraction of the price. So if we now come back to how we want to design master parent derivatives, the way we went about doing this is we started looking at single amino acid substitutions of the peptide. So really what we started doing is just taking, for instance, an arginine and inserting it at position 1 in the sequence. And then we start doing position 2, position 3, position 4. And we ended up doing this for almost all of the amino acids that are available to us. And we generated all of these sequences substituting every residue in master parent. We then got the peptide delivered to us. We made stock solutions of the peptides, and then we screened them against cancer cells. And we also screened them against our normal cells. So in this case, we use PBMCs. And so this way, we're hopefully going to be able to identify, what are the features that make an anti-cancer peptide versus what ones cause toxicity towards normal cells. So now we have over 215 peptides that we want to start looking at, and we screen them for activity against 3 cell lines. So we did the Jerkat cell line again, we did the breast cancer cell line, and we did the PBMCs. So I'm going to just show you the data that comes out of this. And I know there's a lot of numbers on this slide, but it's very easy to understand. If you just think about gray is bad, red is good. Okay, so each one of these boxes represents a single peptide sequence. And what it is, to read these, you look at an arginine, we would substitute in at position 1 of our master parent sequence. So this box represents arginine at position 1, and then all the other residues are the same. And now that's how you would read the table. And as I mentioned, gray is weak or poor activity, and red is strong activity. And so generating substitution matrices like this allows us to very rapidly see which residues are important or essential for the anti-cancer activity of master parent. Where, for instance, if you mutate any of the residues in master parent with a proline, for instance, you virtually wipe out the anti-cancer effect. So they're all in gray. The other thing that you can look at in these highlighted boxes is just a conserved mutation. So this is actually the parent peptide sequence. You can actually now start to pick out mutations that seem to enhance the activity compared to the parent sequence. You can also start to look at blocks of peptides. So for instance, if you introduce hydrophobic residues throughout the sequence, it seems like that improves the anti-cancer activity. Other things that you can look at is for vertical strips. So for instance, this asparagine at position 2 or alanine at position H. It seems like a lot of residues can be accommodated at these positions. So you can swap them out without having a huge effect. So this now starts to give us a map of which residues contribute to the activity. But what we were really interested in is to seeing if we could actually enhance the selectivity. So now what we can do is compare the substitution matrices of Jurkat cells, or cancer cells, with those of PBMCs. And right off the bat, you see that we, kind of reflected in what we already know. Mastoperin itself is already fairly selective. So we didn't expect to see a huge effect on PBMCs, but what we do pick out are several peptides that sort of, if you mutate them, you actually generate more of a general toxin. So these would enhance the toxicity towards PBMCs. So now we know which peptides we can avoid when we start designing our new peptide sequences, and we can sort of focus in on the ones that are red up here and gray down here, okay? But we wanted to try and come up with a little bit more of a sophisticated way. I'm not going to get into how we did it, but we just quickly developed a selectivity index metric where we sort of compared the level of killing of a derivative versus the parent compound. We ended up finding several peptides that had enhanced killing when we based it on our screen data. And so we used these identified residues to design a smaller library of peptides that we could then purify to 95% and retest them for our activity, our anti-cancer activity. So here now is looking at the toxicity profiles of these now new mastoperin derivatives. And so we've screened them against all of our same cancer cell lines again, but the ones I'm going to highlight for you are going to be against Jurkat cells. So we ended up finding, if you look at the red line here, so that's the lowest concentration that we tested, we now start to see in black some of the peptides where we see enhanced activity or enhanced cytotoxicity at these lower concentrations. MDA, MB231 cells, they're harder to kill. But if you go down now to a higher concentration, you see that many of these same peptides here, again, show enhanced activity to the cancer cell lines. If we look at our normal cell lines, so our PBMCs or red blood cells, you can see that some of these things do also enhance our toxicity against PBMCs. So it is one of the things that we always need to make sure is that you go back and check the results of your screen. But based on this, there were several of these peptides that look somewhat promising. So we took those and we ended up doing a little bit more mechanistic studies on them. And so we looked at PI uptake in, again, Jurkat cells. And so this is just looking at PI uptake of peptide exposed cells. And you can see now that the G2, R7, W14, and S8W14 variants have similar, or even I would say R7 and W14 have enhanced PI uptake when you treat them with the peptide. So that's exciting. It looks like we're enhancing this anti-cancer effect. And so I think we're moving forward on this. I think we've got some peptides here, at least with enhanced anti-cancer effects, and hopefully we're going to be able to show that we can actually have improved their selectivity once we actually start taking it forward in some animal models. So in conclusion, we're showing you a couple of quick stories about how mastoperin is an anti-cancer peptide that exhibits selectivity towards cancer cells, and that this activity-guided design approach that we've used has revealed mastoperin derivatives with improved activity. And now just some open questions maybe for to get us moving towards the discussion section. Just some things that I've been thinking about is whether or not the sequence requirements for a selective anti-cancer peptide be generalized. So is there some magical peptide sequence out there that is a perfect anti-cancer peptide? I can't answer that. I don't know if it's true, if we need to start from a template or not. So that's an interesting question going forward. I'm also interested to know kind of what are the limits of this anti-cancer peptide selectivity. So how far apart can we push this selectivity? How specific for a cancer cell can we make it versus non-toxic towards our own cells? And then the last thing that I've been thinking about as well, this is also related to some of our other work, is just how well do some of these in vitro cytotoxicity assays that we do all the time in labs, particularly peptide labs, how well do they correlate with some of this in vivo toxicity that we see? So for instance, hemolysis assay, something that almost every group will do, but is what level of hemolysis is actually something that's going to correlate to in vivo toxicity. I think a lot of us will just say, well, any level of hemolysis is bad, but you know, something that I really have gotten to think about is whether or not these mechanisms are still in play once you start going into, say, a tumor environment, or how you deliver the peptide, where it's delivered in the body, and that kind of stuff. So with that, I'd like to thank a lot of the people in the Hancock lab, particularly Ashley Hilchey, who is sort of the driving force behind a lot of this work, and a very talented he's now a grad student in another lab, but a summer student who worked with us on this. Collaborators around the world. So Octavio Franco, we need to thank, because he was the one that kind of got us going on the mastaperin peptides. Little picture of the Hancock group from pre-COVID times. And yeah, I'd be happy to take any questions if there are any.
Thank you, Evan. I really appreciate you talking us through your research and some interesting information you found with mastaperin. The one question I had, have you seen any other peptides that had the amidation with or without have that different function? Like it was interesting to see that the amidation on the mastaperin made such a big difference.
Yeah, so we haven't specifically looked at that. For us here, we amidate every peptide that we make, because we found, not necessarily specific for cancer activity, but for most activities that we're interested in, blocking that C-terminal charge really does help, regardless of the activity that you're interested in. So I think we have, there's been some studies out in the literature, and certainly of doing even just amidation versus non-amidation can have a huge effect on the activity that you're looking at. So in this case, it was pretty striking, like how it seemed to be an almost switch between the two. But certainly, I do think amidation plays a role. But it's not something that we look at too often, because we just do it automatically and don't even think about it.
So maybe I can add a bit to that. I mean, in our studies on tritryptophan, we also took along a non-amidated peptide, and indeed it had. And I mean, that was the only change, right? Tri-trip-arg is the one with amidation. Tritryptophan was the one without amidation. If you compare the activities of those two peptides, there's a significant difference there as well. The one that was amidated had the higher activity, and so that's why we decided to go along with that, because we wanted to have the highest anti-cancer activity for our starting peptide when we started designing the other peptides. So we left the amidation in, hoping at some point that it would not really change the selectivity. And indeed, in some of our peptides, we ended up in a good place, but as we discussed earlier. But you know, the amidation is something that we should all keep in mind, because it does have a significant effect on these anti-cancer peptides.
I think anytime you do things to the ends of these peptides, you can have and sometimes unwanted effects too. We did some experiments with targeting peptides linked to anti-cancer peptides, and the activity was entirely different sometimes. they targeted well, but they wouldn't kill the cell.
Yeah, well, I mean, most of us, I guess, do not put an acetyl group at the N-terminus, because we all want to preserve that extra positive charge at the N-terminus, because that usually improves the activity and the selectivity. So I think, you know, most of the effects that we're discussing are probably coming from the amidation at the end.
I see a question here. I'll open it up to anybody who wants to answer. Is there any effect for capping or making the helix little rigid? Talking about the amidation, I think they said. Is there any? Does anyone know anything about capping or making?
I can comment. So I think in this case, it seems like there is an effect that's happening. Now, based on the data that I showed, I think in this case, what happens is you block that negative charge at the C terminus, and then that allows the peptide to more readily interact with negatively charged membranes, right? So you now no longer have that charge repulsion. And so in this case, I think it's actually promoting the interaction with membranes and then that helical conformation that you can get. But as was kind of mentioned by David, I think it's important to realize that even small, these are, we think of our peptides as, strings of polypeptides, but they're relatively small in terms of their chemical structure, and even just small perturbations to that can have huge effects on the overall profile. I mean, I think even if you look at the substitution matrices that we've looked at for mutating a single residue in the peptide, you can actually wipe out, for instance, the anti-cancer effects. So, we think of some of these as minor substitutions or minor manipulations, but overall, they might have huge effects on what you're looking at.
Yeah, minor changes seem to have a big effect in the data that you showed. You mentioned a question about hemolysis and whether it's relevant or not. In the mouse experiments that you did, or any of the other panelists, has anyone seen detrimental effects of injecting the anti-cancer peptides into animals?
So in our animal work, that was Dr. Hilche that did that stuff. So I can comment on based on some of the other peptides that we've worked on. And it seems like peptides, from what we've seen, so I haven't gone into it at all, but we've done a lot of work looking at peptide aggregation and trying to understand maybe what are some of these more in vivo toxicity profiles. So one of our sort of rapid assessments for whether or not a peptide might be toxic in vivo is to inject the peptide into the skin, underneath the skin. And what we can see in some of those cases is some peptides will aggregate underneath the surface of the skin. You get these sort of clumps that will form. And then other peptides, they can actually, lead to local redness and sort of swelling there. And so it seems like, at least in our hands, there's two sort of distinct toxicity profiles. Now that's in the skin, whether or not you inject it into the abdominal cavity or you try and do it in veins or anything like that. I think all of those are going to have differences on what some of those toxicities will be. So yeah, I don't know if anybody else has got comments on that.
It's difficult for me to comment because we have not really done any animal studies. It's not our area of strength, so we would have to collaborate with others in order to do animal studies with our peptides.
I think the animal studies can be challenging too, because if you're looking at anything other than an intratumoral administration, so a systemic administration, you're immediately having to up the amount of the dosage to overcome the sink effect of other negatively charged molecules in the body that soak up the peptide, and that's why we and also proteolytic degradation of the peptide, which is why we tried some D-amino acid substitutions years ago, and they worked very well, but unfortunately it meant that the selectivity pretty much disappeared.
Yeah, well, I mean, one of the sinks would, of course, probably be serum albumin, which is a quite negatively charged molecule. I mean, that's 50% of what we have in blood, right? And we've done some studies where we looked at binding of some of these peptides to serum albumin, and while the binding constants are not super strong, they do bind, and given the large amount of the serum albumin, that would shift the equilibrium in that way. On the other hand, you could say that's positive because now the peptide, when it's bound there, is probably more protected from proteolytic enzymes. So, it's always a balance between something good and something bad. going hand in hand.
There is one question I think that just came through. It says, in terms of future directions, what's the next step in elucidating the potential for HDPs as a treatment for cancer? Would the focus be on one specific type of cancer?
My bias is that no, it doesn't have to be a single type of cancer. We've seen lots of data today where different types of cancers. So we've seen leukemia, we've seen breast cancer cell lines. I did not show our data on prostate cancer cell lines and bladder cancer cell lines, of which we've done 4 different ones. And it looks like what you see in one type of cell you see in another type of cell, not in detail, but by and large, you see very similar effects. So I think, some tumors might be more easily accessible, and that might be a first way to consider where we are going to use some of these peptides or try some of these peptides.
I think the ability of these peptides to kill a diverse array of cancer cell types is a real strength compared to conventional chemotherapy, which, oddly, in tissue culture studies, drugs that work well against most cancers only work against 1 or 2 in human beings for reasons that aren't clear.
Mm-hmm.
So if you could have one peptide treats all off the shelf reagent that you could use for different cancers, I think that's a plus.
There's another question here asking, what about targeted therapy for the mastoperin, like using pH-dependent vehicle to release the drug in the tumor?
Yeah, so that's a good question. Actually, I think I saw this question. I thought I'd just hold off because I think it's kind of related to the previous question as well. So I think right now, we're able to find peptides that have anti-cancer effects. We've seen a number of examples today. And I think, going forward is going to be trying to figure out how do we best translate, that activity that we see in vitro in all of our cell line assays and things like that. How do we translate activity to the cancer in patients, right? So I do think things like formulation are really important going forward, not only for anti-cancer effects, but also for a lot of these other properties of peptides that we're interested in. And so seeing if we can overcome some of these issues associated with, off-target toxic effects or aggregation or something like that, can we use some of the knowledge that's out there in sort of drug release and drug release and drug delivery and drug release, and see if we can sort of now get us over that hurdle towards getting them in the patients. And I think also, I'll quickly mention for the previous one, I think another really exciting area is seeing if we can find peptides that actually enhance this immunogenic property towards cancer cells. So I like to think of peptides in terms of their chemical space and knowing whether or not a peptide is lytic or what are its immune properties and things like that, and what are the sequence requirements of those two peptides. And so if we can now start to define what makes a good immunogenic peptide, one that will enhance the immune response towards cancer cells, I think that could be really beneficial. I think if we can also start to define, what makes a good anti-cancer peptide, then all of these things can hopefully come together to actually get us to patients, right? That LTX 315, those results, that's from patients, right? So it's exciting. We know that they work. And I think we just have to now start to get a little bit more creative in how we're getting to the patients and translating some of those.
Yeah, I've looked at the LTX 315 literature. It's a bit ago now, so have maybe not seen the latest stuff, but what is interesting is that apparently it's a very lytic peptide, and companies also call it lytic biopharma. So maybe there's a relationship there. But to make a long story short, I mean, what they claim their mechanism is that it just disrupts the cell, and then all these components of the cancer cell come out and then get targeted by the immune system, and just gears up your immune system towards the cancer. And you know, this is I think it's a cool sort of mechanism, and if we could make this work for all cancers, then you have a lot of potential.
The danger being, though, of you don't want to trigger autoimmune disease to cell proteins that, and that's actually been seen in it's a little off topic, but in adoptive immunotherapy in melanoma patients, where it's working, they lose patches of color in their skin, because not only are the malignant melanocytes targeted, but the healthy ones also get targeted.
That's not good. Yeah. Well, my hair has been changing in color, but that's for other reasons. It turned from red to gray, different than Evan Haney's talk, where.
There's another question here for you, David. It says, I was working with some glioma cell lines. There were some cell lines that produce MGMT, which suppress the chemo effect in patients. How can we fight these personalized behavior of cancer cells during thinking about designing these anti-cancer peptides?
Yeah, that's a good question. I guess we're really, when we're designing these peptides and choosing the most selective and at the same time most potent variants, we're really sidestepping the common mechanisms of chemoresistance. So it doesn't really matter if a cell's overexpressing P-glycoprotein or MDR1 or another molecule that renders a cell chemoresistant, unless something fundamentally unique has happened to their membranes and they've lost their negative charge, the peptides are still going to work.
There's a question here looking at whether there's potential for the anti-cancer peptides to be effective against the superbug bacteria, so probably the resistant ones, considering they disrupt bacterial membrane.
Well, several of us started off in the antimicrobial peptide field and have drifted towards the anti-cancer peptide field recently. I mean, if you look at the amount of time and effort and publications that are now out there for antimicrobial peptides, it's a bit disappointing that there are very few clinical useful peptides that have come forward from that to date, and it's not necessarily clear to me exactly what the reason for that is, and maybe the situation will change in the future, but I hope in the case of anti-cancer peptides that we don't end up in a similar sort of situation where we do all these beautiful basic studies but don't end up with something that has clinical potential. That's my personal bias, having worked on the antimicrobial peptide field for a long time and reviewed numerous papers over those years.
Yeah, I comment on that too. So I think, you know, sort of, as Hans mentioned, a lot of us started in the antimicrobial field. And I should call it the host defense field because of all the activities that these peptides have. And I think there's a lot of there's a lot of hope there that these peptides will be effective against superbugs. Now, whether or not we're going to get like one of these, super antibodies or a magic bullet out of post-defense peptides, I'm not exactly sure. What I would say though is I think if we start to look at them a little bit differently, not necessarily as just like, here's a peptide pill, go take it, right? But can we start using peptides to either enhance the effect of antibiotics, so use them in synergy with each other? Can we target bacteria in a specific growth state? So we've been working a lot on looking antibiofilm peptides, which is sort of a more common growth state of bacteria out in the environment. And focusing on the right activities that would be relevant in vivo. So again, that's why we're also interested in the immunomodulatory effects of these peptides. So, if we're just looking for peptides with an MIC, I don't know if we're actually going to hit on peptides that are going to actually be effective in vivo, if that makes sense, right? So so however best we can sort of start to understand how these peptides might be working in vivo, I think that's going to lead us down the road of developing drugs that are actually based off of these templates.
Yeah, many of these peptides are capable of killing in a test tube MRSA-resistant bacteria and things like that. So obviously, potentially under those circumstances, they work. But how to make that work within an animal model and subsequently a human, I think that's still where we haven't made enough progress yet. David, would you agree with that?
Yeah, I think so. There hasn't been enough. I mean, even going back to the cancer realm, I don't think anyone's used. There've been in vitro studies with chemo-resistant cells and anti-cancer peptides, but inducing a chemo-resistant tumor in an animal and then seeing if the peptide works, I don't think that's been done.
Mm-hmm. Let me ask a question to Valerie. Valerie, I thought it was very interesting that the positive charge in your histidine did not really seem to matter. You must have thought about that. Do you have a rationalization for that?
So the positive charge on the histidine didn't matter for, I guess, which peptide, because it didn't matter for GAD1, but it did matter for the function of GAD2. I think one of the clues is from the molecular dynamics simulations in that I think, a histidine and a histidine, one after the other, might be a little bit like having a tryptophan. Like it's a big chunky thing that wants to be at an interface. Yeah. And so maybe we're a little bit distracted by the fact that its charge does change when you go down to low pH, and maybe it's just more of that's the structure of those two amino acids.
Mm-hmm.
Does anyone else have any other comments or questions they'd like to discuss or ask?
Well, maybe we can end the webinar here, right?
Yeah.
And thank you. Before we go, I want to thank all chairs and speakers. Thank you very much. I hope you enjoyed the webinar, and also thanks to our participant. And then that's all. Goodbye and take care with this pandemic.
Yes.
Bye-bye.
Thank you. Thank you.